My name is Dawn Barlow, and I give Hatfield permission to record and share this presentation. Great.
Okay. So this is
like the thing that in my proposal first told me this, and then once I do that, I'll do all this other stuff.
All right, hello everyone. I have your attention. Thank you so much for coming to our Hatfield seminar series today. We're going to have two speakers. Our first one is Dawn Barlow, and she comes to us as a master's student from Lee Torres' lab, the Geospatial Ecology Marine Megafauna Lab. And we have Rachael Orben, who is also a research associate in that same lab and a research associate in the Seabird Oceanography Lab. So Dawn's up first. We can give her a warm welcome, please. Thanks, Sarah. All right, so as Sarah mentioned, I'm a master's student here at LSU, and I've been working with Lee on this project for the last year. And so I'm excited to share with you guys a little bit about where I'm at this point and kind of future directions for where I hope to take this work. And so my research focuses on blue whales in New Zealand, and as the title suggests, multiple data sources. So this is going to kind of be briefly touching on a number of different angles for how we can study this population. And with multiple data sources come multiple people and multiple collaborators that are involved. So I'll tell you a bit about pieces that various people have worked on. So blue whales were suffered extreme population losses from the commercial whaling industry. This is an image from Antarctica, and these Antarctic blue whales, modeling estimates show that numbers of blue whale populations were reduced to less than 1% of their original size from the commercial whaling industry. And so species conservation depends on robust population assessment, right? So without a current body of knowledge about population status, any conservation efforts will be limited. And so despite that these are enormous animals, right, like literally the biggest, and the fact that they were so severely hunted, still relatively little is known about their population biology, particularly in the southern hemisphere. And so since I'm going to be talking about the southern hemisphere, it's important to acknowledge that there are a couple of different subspecies that live there. Antarctic blue whales, or true blues, which are listed under the IUCN Red List as critically endangered, and pygmy blue whales, and these are listed under the IUCN as data deficient, and that's based on a pretty outdated assessment. And I've included this image here just to illustrate that pygmy is relative when you're talking about blue whales. On your far right, you can see a 45-foot research vessel, and on the far left is a pygmy blue whale calf, which is approximately the same size. And so for the remainder of this talk, I'm going to be focusing on New Zealand, which is our study region here. And so currently, blue whales are listed as migrant in New Zealand waters under the current threat classification system. But also we know that unique calls are produced by the blue whales in New Zealand that are distinct from those produced in either Australia or Antarctica or any other region in the southern hemisphere, really. Which leads us to wonder, is there maybe something unique or different about these blue whales that are found in New Zealand? And so again, coming back to this multiple data sources, this really is an exploration of an understudied region, an understudied population. And so we're taking multiple approaches, including vessel-based surveys of a particular region in New Zealand, looking at opportunistic sighting records. Where have people found blue whales in the past in New Zealand? Looking at acoustics, listening for them, using genetic analysis, and then looking at abundance, how many are there. And in the future, I'm hoping to take this into some habitat modeling. So now I'm going to focus in a little bit more on this region called the South Taranaki Bight, which is right between the North and South Islands. And my advisor, Lee Torres, put forward a hypothesis in 2013 that this is an unrecognized foraging ground for blue whales. And this hypothesis was based on opportunistic records from marine mammal observers. Oh, other direction. Historical records from whaling, from Japanese and Soviet whaling, stranding records, and this, oh, we're advancing real quick there. There we go. This regional upwelling system, there's a plume of cold, productive water that is generated and curves up and around into the South Taranaki Bight region that makes it suitable for blue whale habitat. But there's more going on in this region. It's the most industrially active marine region in New Zealand. There are multiple shipping ports around the South Taranaki Bight, and the major shipping lane of the Cook Strait makes for high densities of vessel traffic. It's also where most of New Zealand's oil and gas industry is concentrated, so there's active drilling and extraction platforms, as well as ongoing seismic surveying for continued exploration. And then it's also the site of an offshore seabed mine for iron sands, and this was actually, the permit was just approved last week, and this is a proposal to extract 50 million tons of iron sands from the seafloor per year over a 35-year period in this region. So as you can tell, aside from the whales that may or may not be there, there's a lot else going on in this region. So this led to the launch of a dedicated study of blue whales in New Zealand waters. And 3 surveys took place in 2014, 2016, and 2017. And so in the field, the most commonly observed behavior, as you can see in this video, is feeding. So here you can see this is captured aerially with a drone. You can see a blue whale approaching a patch of krill. It turns on its side and engulfs the prey patch. And feeding was observed at about 35% of our sightings. And so aside from feeding, another theme is that we observed really high numbers of mother and calf pairs, including this sighting on your left, which is a documentation of nursing behavior, so very young calves. And then there's this racing behavior, which has been hypothesized in other baleen whale species to be associated with competitive breeding behavior amongst males. However, we know from the genetics that both of these animals pictured here are females, so the function of this behavior is unknown. But it's been observed on multiple occasions. So beyond our dedicated surveys, I also compiled records of basically anyone that we trust that has seen a blue whale in New Zealand, right? So this consists of records from the New Zealand Department of Conservation that they keep records, as well as surveys conducted by scientists for other marine mammal studies, for example, where they happen across a blue whale. Seismic surveys are required to have a marine mammal observer on board, and then whale watchers take a lot of photos. Sometimes they can even tell me where they took the photos. And so when I've compiled all of these, I've put together a sighting records for 740 sightings between 1900 and 2017, and 704 of those were associated with precise sighting locations. And when we look at how this kind of plays out, this is showing a distribution, or kind of a picture of how many sightings occurred in which month of the year, and the dark blue portion of these bars are sightings from the South Taranaki Bight region. And you can see that a solid, oftentimes the majority of the sightings take place in the South Taranaki Bight, and that blue whales are observed during every month of the year. And for us Northern Hemisphere folks, I'll remind you that this is the winter time in New Zealand. So you see kind of this dip during the winter, but that there are still sightings during that time. And now if we look at how those opportunistic sightings play out spatially, you can see this is looking at density of blue whale sightings per square kilometer in a 50-kilometer search radius, and you can see this elevated concentration of sightings in the South Taranaki Bight. And what's really important to emphasize here is that a lot of this is opportunistic. This is not necessarily from systematic surveys, so it's not necessarily a true spatial or temporal distribution of blue whales. It's a distribution of reports, and so, or a distribution of effort more. And so kind of the take-home here is that when people are on the water, whether they're looking for blue whales or not, they're finding them during every month of the year. So another tool that's being used on this project is Acoustics. This is Kristen Hodge, and she's a technician at Cornell Bioacoustics program. And what she has there is a marine autonomous recording unit, or a MARU, a hydrophone, and these are deployed in the South Taranaki Bight to listen for blue whales. And this is song, a spectrogram of a song, which to our knowledge is produced by the males in association with breeding behavior. And so 5 of these marine autonomous recording units have been deployed in and around the South Taranaki Bight region, and they will record for a total of 2 years. They're still out there right now. And if we look at the first year of data, what Kristen has done is gone and looked through all of the days of recording to see how often, or to see whether she detects blue whale calls. And so I've put this in this heat map here, and what you're seeing is that the recording unit is on the y-axis and the month of the year on the x. And just a reminder for where those units are. And what first jumps out here is that actually during the winter time, when we saw that decrease in the sighting reports, during 2016 anyway, blue whales are detected almost 100% of the days on most of the units, which kind of supports this, well, maybe the fact that blue whales aren't being seen as often in the winter is maybe because people are not on the water as often in the winter. So another tool we use is photo identification, and this allows us to use the shape of the dorsal fin and pigmentation patterns to identify unique individuals, much like a human fingerprint. And so what I've done with the photo ID is to compile all of our photos we've collected during the systematic surveys and then compare those to any opportunistic sources of photos, as well as existing catalogs of blue whales identified in Australia and Antarctica. And this allows us to find things like this, where this one whale was seen first by researchers from the University of Auckland in November 2010, then by our field team in 2014 in the South Taranaki Bight, then by some whale watchers off of Kaikoura in 2016, and each time with a different calf, each time during a different time of the year. And then 2 weeks ago, I got an email, there's a blue whale in the Hauraki Gulf, and it's the same one. That's kind of fun. No calf this time. So from the photo ID, I've identified 89 unique individuals from our survey effort. And this is a discovery curve, which is showing the cumulative number of identified individuals on the Y and the number of survey days on the X. And what we would expect here is if we're getting close to identifying all of the individuals in a population, that it would start to level off. And as you can see here, it's not doing that, so we're very much still in the discovery phase of this population. And when I combine our photos with everything from everybody else, there's now a catalog of 151 identified individuals within New Zealand, which is the first catalog for blue whales in New Zealand waters. 9 animals were seen in multiple years, and all of those had at least one sighting in the South Taranaki Bight. And no matches were made to any of the whales, any of the existing catalogs in either Australia or Antarctica.
Quick question?
Yeah.
Time period for all the whales in that catalog?
2004 to 2017.
Thank you.
So another tool we can use is genetics. And so here is, you can see a small dart gets launched from a rifle, and that takes a small skin sample. Another source of genetic data are fecal samples. Here you can see Dr. Lee Torres, pooper scooper extraordinaire, scooping up a fecal sample. And then beyond the data that we've collected, there's we collaborate with researchers at the University of Auckland who have an archive of a tissue archive. And so for the next part, I want to give a huge shout out to the Cetacean Conservation Genetics Lab here, Debbie Steele, Angie Sremba, and Scott Baker for this portion of the analysis. And so what Debbie has done is to look at mitochondrial DNA haplotype frequencies and compared our New Zealand data set with published values from other populations that have been studied in the Southern Hemisphere. And when we look at this, you can see that, so it's compared to samples from the Southern Ocean, from Chile mostly, and then from 2 different sources from Australia. And you can see that the New Zealand blue whales are, well, let me draw your attention to first the number of haplotypes shared. For the most part, there's not a whole lot of overlap. The most overlap is with the Australian populations, and that these haplotype frequencies are significantly different between New Zealand and all of these other populations. And another way to look at this visually is using these pie charts. And this, you can see that they're most similar to the Australian population, but that the haplotype the genetic diversity of the New Zealand population is much, much lower. And I'll add that in the literature, it's been kind of established that this Australian population has the lowest genetic diversity of any blue whale population, and that's hypothesized to be due to a small founding population and a small population size. And it seems like this is true to probably an even greater extreme for this New Zealand population. So just to wrap it up, what does all of this mean? Well, we do know that the South Taranaki bight is important for multiple critical life history functions. These whales are feeding there, they're nursing and rearing their calves there, and we have these vocalizations that would imply that they may be breeding there. We know that they're present during every month of the year. And this is from both the sighting reports and the acoustic detections. And this is not necessarily to say that it's the same whales that are there during every month of the year, but there are blue whales present in the South Taranaki bight and in New Zealand all year round. There's no matches to either the catalogs from either Australia or Antarctica. They're genetically distinct from these other regions, and they're characterized by this very low haplotype diversity. So what if it is this unique New Zealand thing? Well, then how many blue whales are there? So to do this, I've come up with a capture-recapture abundance estimate using the photo ID data. And this, to execute this, I used a, there we go, Bayesian Monte Carlo, Markov chain Monte Carlo closed population model using the multi-mark package in R. And so this is of course, a kind of busy model, but just what I want to add here is that this particular package allows for the inclusion of multiple mark types. So I can include both left side and right side photos in the model to get an estimate of how many blue whales there are. And so the first thing I did was to say, okay, during each year of our study, how many whales could have been using that area? How many reasons? So, and what I found was that it's in the 160-ish range that we're probably utilizing our study area during each year, and that it was pretty consistent across the 3 years. The confidence intervals are wide, and that's because of the low number of recaptures. So then, how many are there in New Zealand? That's a trickier question. And so how I approach this is that if we know that most blue whale sightings are concentrated in the South Taranaki bight, they are moving in and out of the South Taranaki bight. We know this from photo ID data, but that most of them are, that oftentimes it seems like most of the blue whales are in this region, that we can treat that as a systematic sample of a bigger New Zealand population. And we have these 3 years of study that we can use as our 3 capture years. And so when I ran that through that same model that was up there before, what I've come up with is around 660 blue whales estimated for this New Zealand population. And so the International Whaling Commission has set out these kind of categories or guidelines for qualifying different abundance estimates. And so here I'm qualifying this as an underestimate or a conservative estimate because we're only sampling from one limited geographic area. So what does this mean? If there is this New Zealand population, if there are blue whales there year-round, we need to really consider what that means in terms of if they're living their whole lives in the midst of this highly industrial presence. And future steps will hopefully be investigating the cumulative effects of this industry presence on the health of this blue whale population. So I've been here for a year. I'm going to be here for a little longer, so I still have plans for where I'm going to take this project next. And kind of the next step is to do some habitat modeling to see, okay, if these blue whales are present in this region, can we predict when and where they're going to be found based on the habitat, based on the environment. And so to do this, I'm hoping to incorporate the locations of these blue whale sightings, potentially behavior at these sightings, with oceanographic data such as temperature and salinity and depth of the mixed layer, as well as prey data from hydroacoustic backscatter, so looking at density and depth and size of their prey. And this will hopefully elucidate some of the interesting trends we've seen in terms of interannual differences. This is satellite images of the region in our 3 different study areas. 2016 was characterized by very strong El Nino conditions. And, oh, those white circles didn't show up too well, but that's kind of a qualitative where most of the blue whale sightings were concentrated. And we did see kind of a shift in where they moved during that really warm year. So hopefully some of these patterns can be teased out in the habitat modeling in the future. And with that, I would like to thank the numerous people who contributed to all of the field work, as well as the many, many scientific collaborators who worked on various parts of the analysis, and the numerous funding agencies. I think there's time for a couple of questions. Yes, Greg.
Iron can sometimes be a limiting nutrient in marine systems. And there have been folks who have even considered adding iron to increase productivity in certain places, with the potential for mining iron in that area. Have you thought at all, I know this is kind of peripheral question, but I'm just wondering if you've given any thought to potential effects that might have on productivity for the food sources in that area.
Yeah, that's a really good question. So the question was, with the potential for the seabed mine for iron sands, could the removal of iron from the system have an impact on primary productivity in the region?
It's not a very perfectly clean operation.
Right. Yeah. So I can't necessarily speak to the like removing iron from the system, how that will affect primary productivity. One thing that entered into a lot of the discourse around the permit application is that there's going to be there will be a sediment plume. So basically, they suck up a bunch of the seafloor, and then there's a magnet that extracts the iron out of it, and then they let the rest of the sand just kind of fall back down. And so that means there's going to be this plume of sediment in the water column, and there's been a lot of concern that that will disrupt primary productivity. And that is not, I'm not sure where levels of iron plays into that.
My thinking was sort of that some of that iron would be resuspended and not completely extracted. I'm not sure if it's in a form that could be readily utilized.
That's an excellent question.
Active and that sort of thing.
Sure. No, that's an excellent question, and I don't have a complete answer for it. But yeah. Yes.
Your acoustics, how far away would the whales need to be in order to be heard?
Yeah. So, given our recording units and the question was how far away can our acoustic recording units hear the whales from? Given the source levels of blue whale vocalizations that have been measured in other populations, and our units, the estimate is between about 70 and 150 kilometers, I believe. So that means that they can be detected from quite a ways away. But one question that I've gotten before is like, well, how do you know you're not just hearing whales from Australia? And so the whales that we would be detecting on our hydrophones would be within the New Zealand exclusive economic zone.
And are they unique? You mentioned that the calls or the sound was unique as well. So that also is evidence that they're not?
Indeed.
So, I think Greg asked my question, which was one of my questions. Which was, so do you identify a particular whale with a particular call?
An individual whale by a unique call? No.
Has
So,
anybody tried to do that? The power of that would be
right, that
enormous.
would be enormous. That's a little bit outside of my field to answer, but I know that I think there has been more effort on that in odontocetes than in baleen whales. But yeah, of course, it would be tremendously useful to say that call came from one whale and that call came from another whale, because as it is, we can say, okay, they're vocalizing on every single day, but it's not necessary. It could be like a really loud whale that's hanging out all the time, right? Or there could be whales on the days that we don't have vocalizations. There could still be whales there that aren't making noise, right? So, but as of right now, there is no way to tell unique individuals from vocalizations, at least in blue whales. Yeah. Yes.
When you do the photo ID recognition, is that a computer program, or are you sitting there identifying it?
This program right here.
Accurate?
Sorry?
Are you fairly confident that that's...
Yeah, and we have a whole series of criteria that have to be met in order. So there's first initial quality control. There have to be a certain number of well, first the photo has to show a certain part of the whale, right? With blue whales, I've realized I just have to have the dorsal fin in there because they're so long that if you just get a section of back, you don't even know which side it is or which part of the whale it is. So there needs to be a dorsal fin in it. It needs to not have a lot of glare on it to where you can actually recognize markings. It needs to not be blurry. And it needs to not be off angle, because that can really mess with you too. And then if I do think I find a match, then there need to be more than one thing that I can match between, that usually I do 3 unless it's a giant tear in the dorsal fin or something like that between individuals. And then we have multiple people look at it to corroborate that everyone agrees that this is a match.
How long does that take?
A lot of hours. Yeah. Yeah.
So when you said there's been no matches found between the whales in New Zealand and whales in Australia and other locations, does that mean you've looked at every whale in Australia, or just so far you haven't found a match?
Yeah, that's a good question. I'm not being very good about repeating the question. Just because I haven't found any matches to Australia, does that mean I've looked at every whale in Australia? So there have been some long ongoing studies of blue whales in a particular region of Australia, and they have their catalog goes back to, I can't remember the year, but they have a long-running photo ID catalog, and they have well over 200 individuals identified, and it really is kind of established that that population is pretty well studied and pretty well identified. And so that's not to say that I have looked at every photo ever taken of any blue whale, but I got kind of the cleaned version of the most up-to-date catalog for Australia and compared that to all of New Zealand.
Well, saying no is a whole lot easier than saying yes.
Yeah.
Right? I mean, you can click through a bunch of photos and say it doesn't match this one relatively quickly. But saying that it does match this one is a lot harder, I think.
Yeah. Yeah. Sure.
And the Australian population, Australia is a big area, and obviously these are very mobile animals. Can move a lot. But I'm just wondering, which area in Australia? Oftentimes, some of the baleen whales, they're thought to be distinct populations on each side.
Yeah. So there's a feeding area in this place called the Bonnie Upwelling, which is kind of southeast Australia, and then and that's where most of the IDs that I looked at came from. There's also been some on the west coast as well. Yeah.
Are there any matches between the east and west coast?
I did not do that part. Yeah, no, that's yeah, no, I didn't compare catalogs between the Australian catalogs. I just compared them all to the New Zealand ones. Yeah. No. Rachel.
I can tell you what our opposites.
her down.
Yeah. Cool, we'll talk later. All right.
I know.
Well, thank you.
I'll show it.
Go for it.
Once you go ahead, I have one more question. But you want to beer one after ultimate?
Oh, I don't know.
Okay, because I can't go to ultimate, but.
But you have more questions.
Think about it this way, Donna. If it's a beer one, you might be able to get them to buy you a beer to answer the question.
What button do I push? All right. So we're going to move from marine predators that move under the water to those that move over the water. So I'll be talking about ontogenetic changes in at-sea distributions of immature short-tailed albatrosses. And the first thing I wanted to mention is I've been brought onto this project at sort of the tail end of it. So as you'll see, much of the research was done before I joined this project. And it was an international collaboration between researchers at the Yamashina Institute for Ornithology and then the Seabird Oceanography Lab, Rob Surian here at Oregon State. And the data set of the distributions of the immature albatrosses was initially part of a master's thesis by Amelia O'Connor. And then more recently, I've added some additional analysis. So we're talking about seabirds. When you're a juvenile seabird, you've never flown before, you're heading out to sea for the first time, how do you know where to go? You're faced with this open ocean, especially as an albatross, you have the capacity to cross that ocean. So how do you choose? So you might have, you might have some innate cues. You might have a compass orientation that sort of tells you that you should go in a certain direction. You might have a preference for a wind direction, so that might help you choose where to go. And you can also learn. So you might see other albatrosses heading in a certain direction, other juveniles are fledging at the same time, you could follow those. And you can learn through experience. So if you find prey or you smell a prey and that was a good experience, you had a good meal, you might look for that cue again and find your way across the ocean. But one thing with seabirds are long-lived individuals. Have any of you heard of the albatross named Wisdom? No. One, the seabirders in the corner. So Wisdom is a Laysan albatross that is, I think, over 65 years old. She nests on on Midway Atoll in the Northwest Hawaiian Islands, and this past year she reared another chick. So she's the oldest banded bird in the world, I think, and she's probably not the oldest albatross in the world. The others just aren't banded. So these birds can live for a really long time, and as an adult, you would have really high survival. But as a juvenile, your survival is much less. So you might have a 60-70% chance of reaching the next year as a juvenile, but as an adult, you might have a 90% chance of living another year. So for juveniles, maybe you fledge when you're really skinny. You might have trouble learning to fly or forage. That could be a source of mortality. You could be caught by fisheries as bycatch. You could be exposed to contaminants. So this is a picture of a Laysan albatross that didn't fledge, that's full of plastic. You could face predation. This is kind of an extreme case, but this is a fledgling black-footed albatross that fledged from the colony and is on the water, and it's sort of challenging to take off, and the sharks know that, and they come and have a feast. And then also you could have trouble, or foraging success could lead to another source of mortality. So if you fledge in a year when prey is not readily available, you might just have a lot of trouble finding food versus another individual that fledged in a year when there's lots of food and you can kind of get by a little bit easier. So basically, juvenile seabirds face a number of risks, and it's a very understudied life history stage. So, I said I'm mostly going to talk about the short-tailed albatross. So this is a picture of a number of individuals. The short-tailed albatross is the largest of the North Pacific albatrosses. Their wingspan can be up to 7.5 feet. They weigh about 25 pounds. And it takes them about 12 years to reach adult plumage. So the birds over here, they're kind of tucking their heads. These are adults, and actually the two that are standing up still haven't reached their adult plumage yet. And the short-tailed albatross is listed by the IUCN as vulnerable, and it's listed as endangered under the Endangered Species Act. And like most albatrosses, it sort of has a long breeding period. So eggs are laid in October, 1 egg per nest, and then the chicks hatch in December, and then they don't fledge until May. So it has a very long chick rearing period. So this is a map of past and current colonies of short-tailed albatrosses, and this is, see that we're in the western North Pacific. Japan's on the top. And we're really going to be focusing in on Torishima, which is shown in the green. And this is the story of the short-tailed albatross. Just like the blue whale that we just listened to, the short-tailed albatrosses were the subject of human extraction. So this is a picture on Torishima sometime in the late 1800s. And it's estimated that about 5 million birds were taken off of this island. And the population was essentially decimated across all of those colonies that you can see here. The birds were harvested for their feathers. So they were made into pillows. Their flight feathers were made into writing quills. And their bodies were also made into fertilizer, and their eggs were harvested for food. So they used all the parts, and they killed most of the albatrosses. Luckily, there were a few that did remain. So after World War II, on Torishima in 1954, there were at least 6 pairs of short-tailed albatrosses left. And this is the population trajectory since then. And now there's somewhere over 4,000 birds. So they're on the upswing. So it's pretty amazing that we still have them. So because they're on the endangered species list, there's some criteria for delisting and certain conservation efforts that can be done or have been done. So the total breeding population, population growth rate, and then considering colonies other than Torishima. So this is a picture of Torishima, Japan. It's smoking, so it's an active volcano. And it's not necessarily the safest place for a colony of seabirds, especially since it's one of the only colonies for this species. So there's Torishima, and then there's another colony on the Senkaku Islands, which is currently contested between Japan, Taiwan, and China. And that one hasn't been visited since 2002. So between Torishima and the Senkaku Islands, there's a lot of uncertainty, and it was decided that another colony was needed for this species to be moved off of the endangered species list. So in summary, the total breeding population is just about there in terms of delisting, or at least lowering the listing from endangered. Population growth rate looks really good, but another colony site is needed. And in a prior, I guess, I don't quite know when this whole effort started, but in 2008 it was decided, or it was initiated to translocate chicks from Torishima to an island in the Bonin Islands, 250 kilometers to the south. So this was done to start a new colony of short-tailed albatrosses, and at this time it hadn't really been tried for other albatross species. So they ran a couple trials with black-footed albatrosses and Laysan albatrosses, tested handling methods, all those things to make sure that their efforts with the short-tailed albatrosses would hopefully be successful. And I have a few pictures. So chicks were translocated from Torishima. They were carried in special heat-controlled boxes. They were hiked out of the volcano, put on a helicopter, and moved to Mukujima, where they then had to be hand-reared. So people had to feed them squid and food through the rest of their life until they fledge. And so here's a picture of them in their, essentially their flower pot nests on their new colony site. And they were moved here, I think around 40 days of age, because the chicks, what was thought is that the chicks needed to be in their new place so that they would essentially imprint on their new colony and hopefully return there. So they brought them there at a pretty young age so that they would hopefully come back. And the question is, are these chicks going to survive? And then, of course, will they return to this colony? So that's the whole point. There's a lot of effort to make a new seabird colony, and especially with a bird like the short-tailed albatross, it might take a really long time. So to answer these questions in terms of will these birds survive, will they return, it was decided to use individual tracking devices. So birds were outfitted with GPS PTT tags in order to, and then this was done with both translocated birds and naturally reared fledglings. And this individual tracking data was used to look at post-fledging survival, and then migration routes, where are these birds going, and then generally overall distributions. And the tags that were used were 22 grams, fairly small. They're transmitting GPS locations every couple of hours, and it was not all of the translocated fledglings were tagged, so some birds had tags attached with tape, and some birds had tags attached with harnesses, and these two different methods allowed for different durations of tracking. So the tags that were attached with tape lasted about a month, up to 8 months, and then the tags that were attached with harnesses lasted anywhere from 43 days to almost 5 years. So it's a much more permanent attachment. And the tags themselves were solar powered, so they can continue to recharge themselves. So what happens when these birds leave the colony? We're able to use the data that the tags send back to assess initial mortalities. So the panel on the left is, there's albatross speed in the blue, and then distance from colony in the red. And you can see that that bird starts moving away from the colony when it fledges and then kind of picks up speed eventually. And you can contrast that to the panel on the right, where the bird doesn't really pick up any speed. So the first bird reaches what we call sustained flight, and the second chick fledged, ended up in the water, ended up on the beach, and then died at some point and drifted out to sea. So this tagging effort allowed us to assess just this general first mortality, and it turned out that 80% or 87% of the hand-reared chicks survived versus 84% of the naturally reared chicks on Torishima, and this was not different between the two. So that was a really nice result, that both groups of chicks were making it to sustained flight. But then what happens? This is the black box of seabird ecology. It's like, where do the juveniles go? What do they do? So just sort of as an aside, these transmitters are really at the cusp of the technology. They're 22 grams, they're quite small, so they're not quite perfect yet. And they're solar powered, they have a small battery, and some of the tracks have gaps. And there was one bird that wasn't heard from for 105 days, and then all of a sudden the tags started working again, sending data. And the birds with harnesses had gaps, oftentimes greater than 2 weeks. So we've learned a lot with these tags, but it's still not, it could be better, right? It's still at the cusp of the technology that we currently have, which is kind of cool. So where did they go? This is the entire data set of where the juvenile short-tailed albatrosses went. There's over 60,000 locations, and the colonies are highlighted in the pink dots on the lower left side. You can see they pretty much traverse the entire North Pacific. Technically, they use waters from the tropics all the way to the Arctic, even though they spent most of their time in the subarctic and Bering Sea. So this is a 50% utilization distribution, so it's where 50% of these data points are. And it's really quite remarkable in this hodgepodge and spaghetti that this utilization distribution is where it is. And it makes perfect sense. When I take the tracks away, you can see that's really highlighting the continental margins, the Aleutian Islands, the Kurils over here, between Japan and Kamchatka, and then also the Bering Sea shelf. And I think later in the presentation I have a picture of a map of adult tracks, and I'll talk a little bit more about this then. But for now, I'm going to step back and talk about the first 15 days. So these chicks have fled from their islands. The naturally reared are in the darker pink. The translocated birds are in the lighter pink. And they've reached sustained flight, so this is their first 15 days of flight. And it's kind of, it's kind of a spaghetti mess. But in general, these birds are heading to the northeast. And I wanted to know, are the birds from Ukujima, which is the translocated site just to the south, 350 miles away, do they adjust for their new location? So in a lot of classic studies of bird migration, they take birds, they move them somewhere else and see if they can find their way home. And in this data set, it's interesting, the birds from Ukujima actually adjusted their course a little bit more to the north, which suggests that they're adjusting for their new colony site and kind of ending up about the same place that the birds from Torishima did. And by August, which is sort of just before the majority of the tape-attached transmitters started falling off, 81% of these birds reached the Bering Sea, which is where adults departing on their non-breeding migrations tend to go. There was one bird in the data set that never went to the Bering Sea, which I found really interesting. And I think that bird was tracked for about 3 years. But everybody else who was tracked long enough to make it to the Bering Sea went there. So how does the distributions compare between these naturally reared and translocated chicks? And this is across 2 years. It's just data from birds carrying transmitters with harnesses, and this is just the core, the 50% utilization distribution. So remember, these birds went all over the place, but this is where they spent most of their time. And you can see that there's some variation in the core regions, and in fact, the Mukujima birds, the translocated birds, have slightly larger core regions, but they're focusing on the same general areas. So again, the continental margins, the Aleutians, Bering Sea shelf break, some of these same places. So this is a really good result because it suggests that these birds they certainly have the capacity to cross the ocean, and shifting their colony location by a couple 100 kilometers doesn't seem to be a very big deal. So from here, I'm just going to talk about some seasonal differences, and we're not really going to talk about differences between these two groups anymore. I'm going to combine the birds because they essentially went to the same place. So this is, again, 2 years of data, the first 2 flight years, and we have summer, autumn, winter, and spring. And what I want you to pick up from these slides is that these birds are sort of moving across the North Pacific depending on the season. And they're spending summer and autumn. There's high concentrations of birds in the Bering Sea, and also along the Aleutians. In the winter, that's when you might see a juvenile short-tailed albatross in the California current. And then in the spring, they move into the Gulf of Alaska and on the Aleutians. And one thing that struck me is that in all of these 4 seasons, there are high concentrations of short-tailed albatrosses in the Aleutians. And to some extent, you could say the same for the Kuril Islands. And the Aleutians are really known as a hotspot for multiple seabird species. This is a photo of a short-tailed albatross in the eastern Aleutians, and you can see there's Lake Sand albatross, there's some shearwaters. These passes also are known for high concentrations of fulmars, other surface foraging seabirds. And it's thought that tidally driven currents really bring the prey to the surface for these surface foragers. So perhaps it's not surprising that these birds are ending up in the Aleutians year-round. So before I showed you, I'm showing you these utilization distributions that really highlight the continental margins, the Aleutians. How does this happen as the birds go out and move across these different areas? And so this is the time spent in 4 different bathymetric domains. So we have the shelf, less than 200 meters, shelf break, the slope, and then oceanic waters, the deepest. And you can see as they age across year 1, 2, and into their 3rd year, birds are spending more and more time at the shelf break and the slope regions and less time in those oceanic waters. And this is a picture or a map of the juvenile. Some of the juvenile data is not the whole data set in orange, and then the tracks of adult birds tracked from their colony on Torishima in the red. And you can really see that there are some differences in these juvenile distributions. So the juveniles tend to explore much greater extent of the North Pacific. So they're in the Sea of Okhotsk, they're along the US West Coast, they're exploring farther north into the Bering Sea. And then you can compare that to the densities of the adults in these continental margin areas. See, as the birds are aging, they're kind of honing in on where the adults are spending their time. And I was kind of curious about this, whether birds were sort of going back to the exact same place. Like, if I'd been here last year, am I going to go here again this year? Or are they really honing in on that habitat? Like, this is the right kind of place. I don't really care that I've been exactly here before. And this is tracks of 9 of the birds that were tracked for full multiple years. And you can it's a bit of a spaghetti mess, but you can kind of see the variation in these tracks. But also you can see that they are returning to some of these places. And this is GPS data, so it's accurate to 10 meters. So you can really know if a bird went back to the same place, right?
Is the starting year the same for those numbers?
It's not. So
Okay,
it's
okay.
just, they're colored by flight year.
Okay, thanks.
So, year of bird. So here's a sort of a depiction of the next analysis that I'm going to show you. And what I really wanted to know is if birds were coming back to the same place. And so I calculated the distance between all of the points on the between a track of year 1 versus year 2 to see if in year 2 if they returned to places they had been in year 1. And I didn't know what scale this would really be relevant at. So it's like, you know, are you home when you come back to the town you live in? Are you home when you turn the corner into the block that you live on? Are you home when you open the door? So like, what spatial scale does it really matter for an albatross? Like, I don't know. So instead of trying to guess, I just did this at multiple scales. So across all the tracks, and then at increasing scales. So this is a plot of, with X is the scale. So if you're within 25 kilometers, then you're assumed to be site faithful. If you're within 50 kilometers, you're assumed to be site faithful. And you can see that in year 3 versus the previous 2 years, they're a little bit more, they're spending more of their time in places where they've kind of been before, right? That's what you'd expect. They've had more chances to be there. But, you know, is this more than, are you more site faithful than would happen by random chance? So then I randomized all the tracks and created a random, a similar random curve to see if, you know, maybe they just happened to be there and they could have, it could have just happened by chance. And in fact, they're very, very close. There's only one scale in year 3 versus year 1 and 2 where it seems like maybe at 20 kilometers they could be more likely to return to somewhere purposefully rather than what would happen by chance. But it's interesting that even at year 4, they're spending about 52% of their time exploring new areas. So they're not set in their ways by any means. They're going to new places throughout all these years. And I think all of the really sort of interesting, sort of outside the box movements in that plot that I first showed you, there was one bird that traveled into the tropics, one bird that went over towards Mexico. Those were done by third-year birds. So they're not set in their ways by year 2. They're still exploring into their 3rd year, and potentially more. We just don't have the data to look at it. So this is the where are they now sort of slide of the presentation. So these birds have traversed the North Pacific multiple times, but some birds started to return to the colony at 3 years old, when they were 3 years old and began pairing. So on the upper right-hand corner of the plot, you can see the years starting in 2008 at the beginning of the translocation effort, and the number of birds attending this new colony site. And by, I actually forget the year, if it was 2016, but the first hand-reared bird returned to the colony at 8 years of age and was able to raise a chick. So this is sort of the first, the establishment of this new colony. It's very small at the moment, but hopefully it will continue to grow. And it's a wonderful result considering all the effort that was put into moving these chicks to this new place. So thinking about sort of the juvenile movements that I just showed you, some of the conservation implications of these is that these young short-tailed albatrosses are spending time in the exclusive economic zones of Russia, the US, Canada, Japan, and even Mexico, to a much lesser extent. And they could be vulnerable to fisheries in all of these waters. And the juveniles were much more widely ranging than the adults, and they explored nearly all of the previous range that was sort of historically thought to be part of the short-tailed albatross range. There's reports from Bering Strait in the mid-1800s of seeing lots of short-tailed albatrosses, and that went away with human extraction. And then I think that this wide-ranging and sort of exploratory capacity of these individuals may have helped them to be really the dominant albatross of the North Pacific. And it could also be helping to contribute to the faster than expected population growth. So that population curve that I showed you at the beginning is a little bit faster than one might expect for these long-lived birds. So perhaps it's their ability to be really flexible and use a lot of the North Pacific. So remind you that, again, I was really just a spokesperson for this project, and so much effort went into essentially making a new seabird colony. So I'll take any questions. Jody.
Do you know if the original 6 pairs or the original 6 females kept coming back and repopulating? In that growth curve that you showed, you said it started with six nesting pairs.
Mm-hmm.
Is there genetic analysis to back up how much of that population growth originally came from that stock?
You know, I'm not sure how often that island was visited by biologists and how carefully those birds were watched. I don't know. There are some interesting genetic things with short-tailed albatrosses, so I sort of glossed over. The bulk of the population is on Torishima, but then there's another population on the Senakaku Islands, which is actually known to be genetically somewhat distinct. And that's not, I guess, uncommon for large albatrosses. You have the southern and northern royal albatrosses of New Zealand that are considered to be, I think, 2 different species. So there's some, I guess, genetic diversity that has remained in this population.
Well, I'm just wondering about that. Also, that one that didn't ever go to the Bering Sea suggests that, you know, they might have general patterns, but the way that you get genetic spread there, just as we heard in Shantae's talk this morning about salmon that most of the time go back to their stream of rearing, right? Sometimes don't. Whether they could hop islands and maybe rear the next generation on a different island.
I think, I don't quite remember all the individual details, but I know a Senakaku bird has been, maybe it was one of the birds that paired on Mukujima or has been seen on Torishima. There are some sightings of Senakaku birds on those islands. And then they're also caught in fisheries bycatch as well. So there's some mixing over the large North Pacific.
Mm-hmm.
Don.
So the foraging site fidelity metric that you developed, has anybody done something similar to compare adults to juveniles in any species? That's pretty cool.
As far as like
how much they explore new areas year after year, right? You were still seeing that in the juveniles, by year 3 of tracking. I wonder how much adults explore new territories from one year to another.
It's true, and I'm not sure. I think there's been a lot of work with sort of foraging trips when adults are attending chicks, so they're central place foraging, attending the colonies, so they're really spatially constrained. But there's been less long-term tracking of adults on migration. I think I've seen some work at conferences, but I haven't seen it published yet.
I've got a question for both of us, I think relative to both of you. Has there been any work done that you're aware of on photogrammetry from satellite of krill? Because that image that you showed, that video that you showed was killer. That
Yeah.
That was amazing.
Well, stay tuned.
Yeah.
Lee and John Burnett, who's just finishing his PhD in forestry department, he developed a lot of photogrammetry methods for measuring whale morphology, morphometrics, basically.
Mhm.
We're working on doing a similar thing for that drone image where you can't always see krill in patches that discrete, and that time it was actually like at the surface. Oftentimes the patches are not at the surface. But we're going to try and use some photogrammetry to measure patch size and then like how the whale results it
Well, but the surface patches are certainly relevant to
to the albatross.
the albatross because they're going to bring larger. What do albatross eat?
All sorts of things.
Okay.
They eat squids. There can be scavengers, so they can eat dead. Short-tailed albatrosses have been seen eating dead whales, fish eggs,
Okay.
fish,
Mhm.
everything.
Yeah. But the scale you're operating on is one that is now getting into the satellite regime, and so if you can get satellites that are in geostationary orbit, then you have access to imagery like that. That imagery is pretty fine resolution.
That should be pretty tough to get it up there. There's so much cloud cover and that to be able to get,
Okay. Yeah.
krill patches and things like that. I think it'd be tough, but I don't
Okay.
know for sure. I'm
Someday.
just guessing.
Yeah, but it does sort of point to another black hole, like what we don't know about short-tailed albatrosses. There's very little known about their diet when they're crossing the ocean. Like, they must be switching their prey species, right? They have to be eating something different when they're in the ocean versus off the west coast versus the Krill Islands, the northern Bering Sea. And yeah, we just don't know. And we can guess, of course, but.
We need an army of young undergraduate minds who can tackle imaging challenges.
True.
That are well funded.
Yeah.
You're not cheap yet.
Does anybody have any more questions, or should we? I smell pizza.
I'd like to thank both of you for great presentations. Really, really great and interesting stuff.
Thank you. Well, thank you guys for listening.
All right, so announcement for our grad students and interns. Next week, 5-8 grad panel. We'll have some grad students come in. More of them will be coming in around 5. They're in class right now, Spanish class, so when that ends, they'll come in. But in the meantime, we have pizza for you. So we'll lay that out so you can grab some slices of pizza, eat, kind of mingle a bit, and then right before 5 or around 5, we'll open the floor for Q&A about graduate school. So help yourselves. It's down there.
Alright, I'll end that meeting.
So,